Hi, everybody. This is Alan Russell from Edgewise Therapeutics, and I'm going to be starting this presentation where we'll be talking about muscle health and exercise in Duchenne muscular dystrophy. Duchenne is a muscle disease that results in muscle injury, and I think it would be helpful to all you folks just to understand exactly what happens to muscle, how exercise is useful, and how you might adapt exercise for better outcomes. So there'll be three of us talking in this presentation. I'm Alan Russell. I've been around for quite a few years in the drug discovery business for muscle therapeutics. I've been at both Cytokinetics and GlaxoSmithKline over on the East Coast of America, making muscle therapeutics. Recently, I started a new company, Edgewise Therapeutics. We've been going for 3 years, making novel medicines for Duchenne muscular dystrophy, and hopefully we'll talk to you later about those. So I'll be going first, just introducing muscle physiology and what happens to your muscles when you exercise. Tina Duong from Stanford will give the next part of the presentation, where she introduces kind of that connector between the physiology and the whole body exercise benefits. So just understanding why exercise is good. And then Donovan Lott will close out the presentation. Donovan's from the University of Florida, and he will close out the presentation with some practical data on exercise in Duchenne muscular dystrophy. So I just told you all of this, so I don't need to go over it again. I'm going to give you the overview, then we're going to do the bridging, and then Donovan will close it out. So just a few interesting facts about skeletal muscle. I think it's often an organ that we take for granted, but it is one of the most important organs in the body. It's certainly the largest. It's about 40% of your lean mass. I think we're used to seeing people of all sorts of shapes and sizes in our lives. And I've got two people illustrating the range and diversity of skeletal muscle there. So you've got an endurance athlete on the left, very, very skinny, but able to use those muscles for impressive lengths of time. And then you've got a power athlete here on the right, who's of course much beefier. These muscles are really essentially built for strength, but if he goes for a run, he's probably not going to do too good. As we all know, skeletal muscle is essential for coordinated movement, and that's mediated through the contractile function of the tissue. But it's also one of the biggest organs for glucose uptake. So your body metabolism in many ways is dependent upon the health of your skeletal muscle, and we'll touch upon that later in the presentation. And then, as I inferred with those people above, it has the most amazing ability to adapt to a wide variety of stressful situations. If you think about how the weather changes, for instance, and we go from extreme heat to extreme cold, it's amazing your muscles can adapt to that, just keep you going. And that's our inner caveman there driving us along to get to that next physical function, allows us to move around, but it also does two other important things. It consumes energy in the form of adenosine triphosphate. It's a high energy phosphate, but in order to do that, it needs to suck up a lot of nutrients. As it's 40% of your lean mass, you can imagine that's quite a lot of hungry tissue that needs feeding. And then the third part of this triangle is that it grows and shrinks in response to your nutritional and exercise status. So all of these three functions work together in order to maintain healthy muscle. Of course, there's a fine balance there between growth and loss. Skeletal muscle is an extremely energy-hungry organ, as I just mentioned, and the body would, in many ways, rather you didn't consume all of that energy. So whenever it's given an opportunity, the body will get rid of muscle and use the energy liberated from the loss of muscle to feed your other organs. Even resting muscle takes up about 20% of your resting metabolism. As I'll show you on the next slide, certain events can really trigger amazing amounts of loss. So here's my favorite example of loss. It's a study from some clinical scientists in London where they measured muscle cross-section, so how thick your muscles are when you go into intensive care. So this study took quite a large number of intensive care patients who were admitted for all sorts of things wrong with them, and then studied how their muscles changed over a 10-day period. And what you can see is the cross-sectional area, so the diameter of the muscle essentially decreased by almost 20% in just 10 days. So just 10 days of lying in an intensive care unit is enough for you to lose a fifth of your muscle mass, which is kind of a terrifying thought. What the body is doing with that is it's dissolving the energy in the muscle, and it's releasing that into the bloodstream to feed in organs that it views as more important, such as the brain. This happens to all of us when we're inactive, and it's an important reason why muscle use is so essential to overall health. Of course, there's good news too. There's positive adaptation with exercise, right? So in extreme cases of disuse, like intensive care, muscle goes away, but when you exercise, muscle grows, of course. Now, there's three basic types of exercise, and they're often termed by their scientific names, which is really why I'm putting this slide up, just because these words get batted around and they can often lead to confusion. So you have fixed length exercise. So that's where you're resisting a force, but your limbs aren't moving. Okay? So in this case, the person has their arm outstretched with a weight. They're not moving up or down, they're just resisting that load. That's a fixed length fixed length exercise, and that's also often called isometric. It's just another way of saying fixed length. The next two exercises are really common in kind of exercise physiology. You have a shortening exercise, which is often called a concentric exercise. So that's where essentially you're doing a bench prep, a pull, right? So you're taking this weight, and instead of holding it straight, you're moving your arm towards your bicep here. So you're essentially doing a curl. That's a shortening maneuver because this muscle is now both contracting and shortening at the same time. And that's what this figure on the right-hand side is illustrating. This is the length change. So the muscle is getting shorter, and when it does so, the force actually decreases in the muscle because the length is decreasing. Okay. You can also do the opposite maneuver. It's a lengthening maneuver. So now you're extending the arm with the weight, and now this muscle is becoming stretched. When you do that kind of maneuver, which is a lengthening or eccentric maneuver, the muscle lengthens, of course, but the force is enhanced. So you can see here, you get this huge spike of force. So imagine you're holding a shopping bag and your arm is outstretched. That's an eccentric maneuver. You're having enhanced force development and enhanced stress on the muscle. That enhanced stress is great if you have healthy muscle and you're doing normal exercise. So what I have here is an example of a paper published back in 2019, where they took healthy older people and did a whole bunch of kind of in-house exercises that were designed to lengthen your muscles. And what the scientists saw compared to the concentric or shortening exercises is lengthening exercises give you a much bigger boost in strength when you do them. And that's because the internal stress on the muscle is greater. So more stress equals faster adaptation and greater strength gains. However, you can always overcook that. As I mentioned, eccentric exercise gives you the biggest internal stress in the muscle. And if you take someone who hasn't done a lot of exercise and you give them repeated rounds of that type of exercise, so this is a lengthening exercise of the arm, a weight is essentially being applied to this machine, and the person is trying as hard as they can to resist that weight. And their arm is being lengthened in the process. So you can imagine their bicep is being lengthened over and over again in this kind of protocol. And after the exercise, the patients essentially have a form of muscle myopathy. They get pain, which lasts for about 6 or 7 days. They have an acute strength decrease. They have decreases in their range of motion, so the ability for them to extend their arm, and their creatine kinase spikes up. So in many ways, these are observations that are quite common to Duchenne or other muscle myopathies, in which the muscle has been exercised to the point of injury, and now that's yielding negative consequences. And that gives me the transition into talking about Duchenne and how the lack of dystrophin alters our response to exercise. So of course, in Duchenne, you're missing the dystrophin elements of muscle. So dystrophin, as you well know, is one of the largest proteins in the body, and it has a really interesting specific function, which is to connect muscle fibers together. So in this case, what we have is two muscle fibers in pink here at the top and the bottom, and then we have the extracellular matrix that surrounds all of the muscle, denoted here in yellow. The dystrophin complex essentially links the contraction machinery to this kind of extracellular matrix. And in doing so, it allows the muscle to transmit force both longitudinally along the muscle, but also transversely between the fibers, right? Because it's connecting all of this stuff together. If this is not present, when the muscle contracts, you get stress. You get stress between the extracellular matrix and the muscle because this is moving now and this is not, and that leads to internal stress. That stress ends up causing hypercontraction and fiber collapse with muscle contraction. So on the left here, what we have is a happy muscle. This is an uninjured muscle, and you can see this beautiful striated pattern of all of the contractile machinery. It's a really organized tissue, one of the most organized tissues. When muscle contracts, either in healthy individuals doing that kind of eccentric exercise or in Duchenne, you get this type of internal stress. Everything starts becoming discordant and messy looking as these filaments all move in a non-coordinated fashion, and that's what you get this thing called streaming. Eventually what happens is that stress causes membrane stress because of the extracellular membrane not moving. That causes calcium to enter into the tissue, and that starts essentially killing the muscle fibers. It causes hypercontraction as the elements overcontract, and that's what's shown here is they're all packing into each other. And then eventually you get internal breakage and collapse where the whole machinery just packs in on itself. At this point, what happens is that the fiber has to regenerate. A signal has been sent to the body that the muscle has died, or that fiber has died. And what happens is the inflammatory machinery has to be recruited to activate your stem cells to build new fibers. And as you're aware, that happens quite healthily up and until age starts catching up with you, and muscle gets replaced by fibrotic tissue, and that's where the problems really start. Just the same as in healthy individuals, eccentric contractions really accentuate the problem in Duchenne. And this is a preclinical study, so this is a study in mouse muscle, but it's very, very similar to patients. What we have on the left-hand side is a dystrophic muscle from a mouse undergoing fixed length contraction. So this isometric exercise, essentially. And then what they've done is they've done two different types of exercise. One is a concentric exercise, so a shortening exercise, and the other is a lengthening exercise. And you can see that under both fixed length and shortening conditions, the muscle works just perfectly fine. There's no decrease in force, it's looking healthy. What happens when the muscle undergoes eccentric exercise is you get this really characteristic decline in force when you do repeated contractions, and that's because of this internal injury. So eccentric exercise, much the same as in healthy people who are overdoing it, is really the trigger for patients to lose function. That function, of course, is associated with other stuff, and I just threw this in because Donovan will be talking about it later. You know, you're well aware of high levels of creatine kinase associated with the disease, But just the same as in healthy individuals doing eccentric exercise, you can actually see a relationship between activity and creatine kinase in patients also. Now, in the case of healthy people, they had to do a very defined exercise over and over again in kind of an unfamiliar way to get the signal. Of course, in Duchenne, because of that additional fragility, normal exercise that includes eccentric lengthening maneuvers will give you a spike in that too. This is a very interesting study from a number of years ago where the researchers tracked 4 days of activity of patients in a controlled way and took blood samples every 6 hours. And what you can see is 2 biomarkers, creatine kinase here in black in circulation. And the patients were at the track doing exercise. They had a bed rest day. Then they went to the zoo, which was the most stressful day, really. And then they were back on the ward for an inpatient analysis. And you can see essentially as their activity went up and down, so did their biomarker. So in many ways, you're seeing a similar kind of relationship between activity and biomarker. You know, quite often in patients, we don't measure this nearly as frequently, and then you don't really know the relationship between activity and biomarker. This study nicely put these things together. So just in summary, muscle, as I mentioned, is a very large organ. It consumes a lot of energy, and under many conditions, it would rather not be as big as it is. So it's quite happy to shrink. It can also grow. The type of exercise that you do modifies how it grows. Of course, that exercise can go in the wrong direction in Duchenne and actually enhance fiber injury, particularly under eccentric conditions. Tina, in the next part of this presentation, we'll tell you about some of the positive effects of exercise and not just the bad news that I've just described.
Okay, great. Thanks, Alan, so much for prefacing us on some of the impacts of exercise on the physiological perspective. I'll talk a little bit about the benefits of exercise, trying to look at the positive, but also trying to understand what gaps that we need to address in our understanding of exercise itself. So I'll start off with the positive benefits of exercise. As you can see here on the page, that a lot of what we're talking about on the impacts of exercise, positive and negative, the impacts are very much relevant to Duchenne muscular dystrophy. For example, with exercise, we do know that there's a decrease in inflammation, which is very prevalent in Duchenne, and methods of treatment and management of that currently as standard of care are steroids. In addition to that, exercise reduces the cellular resistance to damage, which, as Alan mentioned, with the cellular matrix fragility, that's going to be important in Duchenne. And then exercise also decreases oxidative stress that's placed from a cellular level, and improve muscle metabolomics. And what that really consists of is oxygenation of the muscle fibers, and balances the energy level and the cellular level, as well as the contractility or force that the muscle is able to contract, and has to do with the different fiber type and switching that occurs with different types of activity and exercise. But with that, as Alan noted, there's also detriments to exercise. So what we do know is, like in Duchenne, that the dystrophic muscle decreases and shows as decreases in metabolic capacity and increases oxidative stress. But we also know that without the use of these muscles, then you have the exacerbation of atrophy or disuse. And so the confusion then is, what do you do? Because what you definitely don't want is a compound effect of progression of muscle disease on top of disuse. And so the gaps here that we need to address is, how can we find that balance? And so the rest of the presentation, I'll talk about some of the questions that we have as a community to answer this. And there's lots of different ways to bridging this gap on exercise. And so we'll address a few of those on reviewing of what is currently out there, which is not a ton that is known. As most of you know, a lot of the studies that have been done in Duchenne is in mice. So I'll try to de-emphasize some of the mouse stuff, but mice work really does help us kind of understand the physiology or physiological impacts of exercise on dystrophic muscles. I'll start off though with a study that is not done in mice, so done in boys with Duchenne, on an arm-assisted or leg cycling training program. And the design of this study was really to decrease the eccentric contractions that Alan talked about that were more detrimental to muscle. And so this was a low intensity exercise that the boys were they use a cycle ergometer, as you can see here on the right side, in which they can cycle with their arms or their legs, and when they're not able to cycle, there's an assisted motor that helps assist through that. So what they use as an outcome measure to measure the effectiveness, one of the outcomes was a motor function test. It's called the MFM32, and within this 28-week period, they had two groups, boys who did the assisted cycle and boys who were inactive. And as you can see here on the right side, across the yellow line, anything above the yellow line showed improvements in the motor function assessment, and those below the yellow line shows that there were negative impacts. And so you can see here that a lot of the boys who were in the exercise group using this low intensity type of training had positive benefits. And so the outcome here is that low intensity training may contribute to decline and deterioration of disuse for boys with Duchenne. Now that we know that muscle is impacted, dystrophic muscle is impacted, and may have improvements in function, we really need to understand what the possible reasonings are behind it. And it really goes back pretty historically. When Duchenne was first described, there was a debate on whether it should be called a mitochondrial myopathy. The reason being is that the mitochondria has been a key and is a key player in the pathogenesis of Duchenne. So mitochondria is kind of like an energy factory for cells. And so it's the powerhouse of these cells. And what researchers have found is that the ATP level, so the powerhouse of the cell, energy producing piece of the muscle, is 50% in non-dystrophic muscles, which is pretty dramatically less. And then on top of it, the mitochondrial synthesis is also reduced by 60%. So energy level of these muscle cells are half of that of normals. And then on top of it, the ability to make energy is also quite reduced in dystrophic muscle. And all of that leads to decrease or increased metabolic and oxidative stress that's really similar to aging or aging muscle. And so then, of course, when you think about the mitochondria, we can't think about that without looking at calcium. And we know in Duchenne, calcium regulation is quite a problem due to the influx of calcium into the cells. And so the mitochondria is quite sensitive to this influx of calcium, and which leads to the mitochondrial dysfunction and cell death that we see that we would like to try to avoid. And so we can't talk about dystrophic muscles without really addressing that there's a mitochondrial component that is driven and a lot by muscle activation and possibly in exercise. And so then beyond muscle itself, are there other benefits beyond muscle function? So we know that whole body health is very much influenced by exercise. And improvements in this muscle health increases energy expenditure that has other impacts in other organs such as the liver for glucose metabolism, and it decreases adipose or fatty tissue and has really good benefits in behavior or brain health. On top of it, we also can't forget to look at the cardiac and the pulmonary component of exercise. And so there's not a ton of studies looking at how increased muscle activation may impact cardiac function. And so there's a study that we did looking at mice. And so I avoid talking about mice, but the reason why I brought this study up that we did is because we did a study looking at different modes of exercise. So we looked at treadmill, downhill treadmill, which is indicated by DT, horizontal treadmill, and no treatment. And what we found was typically what you've heard before, that eccentric exercise was not great on muscle function, indicated by decreased strength in the mice. However, we found that the light intensity voluntary wheel exercise may have positive effects on cardiac function. And that's highlighted here on the right side. So you could see on the x-axis are the treatment modes: downhill treadmill, which is indicated by DT, horizontal treadmill, no treatment, and voluntary wheel. On the y-axis, you see the percent systolic function representing cardiac function. And what you see here is in the light intensity or voluntary wheel exercise, where the mice could run whenever they felt like on this wheel system in their home, you saw that there's a statistical significance in systolic function compared to that of the mice who didn't exercise and even the mice who did horizontal exercise. So I think there's room for a further deeper dive into the possible effects of light exercise, also not only in helping with motor muscle function, but possible cardiac function. Okay, and so then when we look at exercise, why does exercise really matter besides the question of disuse or muscle deterioration? A lot of times when we look at Duchenne, we look at what are the disease modifiers that may impact the progression of the disease or the severity or the rate of decline in the natural history of Duchenne. And we know that biomarkers, genetics such as SNPs, are associated with certain responders and the trajectory of decline. But the one thing that we don't really quite understand in Duchenne is the lifestyle. With a more active lifestyle versus a sedentary lifestyle, how does that impact muscles' ability to function, and how does that possibly change the trajectory of disease progression? We know that dystrophin is a key structural protein of muscle fibers. And because of that, we know that there's going to be incredible impacts on the mitochondrial function. And so beyond that, we know that in Duchenne, the inflammatory pathways are greatly impacted. So with exercise's impact on the inflammatory and immune system, how would exercise change the downstream effects there? And then, of course, we can't talk about muscle activation without talking about some of the current drug developments that are happening now. We know that with these therapies, it's not going to fix 100% of muscle function. And so can exercise itself have complementary impacts on function in totality? And then, of course, with increased muscle activation, there's going to be questions about the PK and PD of dosing and frequency of redosing for some of these therapies that are being produced. And so with therapies that improve muscle function, how is that going to change the muscle metabolism of drugs that are being developed out there? So there we talk about the gaps. And so when we talk about gaps, we talk about exercise. We forget that we should talk about it in the similar sense of taking medication. What's the dose response to exercise? What kind should we do? How much should we do? And what is its impact on the pathophysiology of Duchenne? And then, of course, looking at the impacts, when you have increased muscle function, as we talked about earlier, there's other organ systems that are impacted, such as the cardiopulmonary system. How does that play in increased muscle function? For example, in Becker muscular dystrophy, there's an increased cardiac phenotype. So is that because there is more muscle function? And that's something that's going to be important work to understand as we try to improve the muscle phenotype in Duchenne. And of course, I talked about muscle metabolism. The PK and PD aspect of drug development is going to be important to take into consideration the increased muscle use and metabolism as boys get stronger with treatment. And then, of course, can conjunctive therapy improve health outcomes or slow disease progression, and what is its role in the variability that we're seeing in Duchenne itself? And of course, we can't talk about gaps without talking about how to measure these gaps. And so we can't understand the benefits of exercise without fully being able to explain what's happening. And so some of the outcomes that are currently being used to monitor exercise includes the toolboxes that you all are very familiar with: muscle testing, including manual muscle testing, handheld myometry, and motor function tests, including the North Star time function test, and less familiar in the US is the motor function measure that was used in the disuse study from Jensen's lab. And so the other way that's really great to measure exercise, that's typically done in athletes, are what's called CPET, cardiopulmonary exercise testing. It's a standardized way to really accurately measure oxygen utilization. And then a few other ways with looking at muscles' use of oxygen is the near infrared spectroscopy, which looks at oxygen extraction in the muscle fiber level, and then magnetic resonance spectroscopy, which is looking at the energetics of the skeletal muscle itself. And so that being said, we'll move on to the later half of this talk, talking about the studies that are going on, but I'll kick this aspect of the talk off with a study that we're doing at Stanford in trying to make some gains in better understanding exercise impacts on the other organ systems besides muscle, and that is CPET testing. So as I said, CPET testing is the most direct way to look at oxygen utilization and uptake in the muscles, but it also allows us to understand the interaction between the cardiovascular and the respiratory system, which is going to be very, very important, not only in the development of exercise studies, but as treatments are helping our young men to be able to move and exercise more. And so we're developing a protocol that's specifically for boys with Duchenne who are ambulatory and non-ambulatory, because the current protocol that's for VO2 may be too aggressive, and boys may not be able to get to their VO2 max. That requires a lot of muscle function. And so we are in development of that standard protocol. In addition, we're using the NIRS device to also assess muscle fatigue. And that's just a device that's on the skin that's really not invasive and not very expensive, that looks at oxygenated and deoxygenated hemoglobin so that you can see them from a fiber level, how much oxygen is being utilized by the muscle fibers. And that being said, I will let Donovan finish us off with what's currently going on.
Well, thank you very much, Alan and Tina. I'm really excited to talk a little bit further about some of these clinical applications that we've done recently when we start thinking about what about strength and exercise in boys with DMD. And we had this initial question, what we wanted to do. Of course, first and foremost, it was, well, who are we going to be specifically targeting with a research study to be able to be a little bit more focused? And of course, this was going to include boys with the diagnosis of Duchenne muscular dystrophy. We decided that from an age range, we would be looking at boys who are at least 7 years of age so that they would be able to comply and follow specific instructions for exercise. And then we had a maximum age of 10, so that coupled with the inclusion criteria of being able to walk at least 100 meters and being able to ascend 4 steps would provide a greater likelihood of having some viable muscle tissue that we felt we'd have a greater likelihood of impacting in these initial investigative studies. And because we all know about the impact that steroids have on skeletal muscle, we felt this was an important factor to take into consideration. And so another criterion was that all of the boys who would participate would already be on corticosteroids as prescribed by their physician. So with these pieces in mind, our overall purpose was to explore the safety and feasibility of a mild to moderate resistance isometric exercise program in ambulatory boys with DMD. And there are 2 studies that we focused in on. And the first one really goes back to what Tina was talking about when we start thinking about exercise from a medical or a pharmaceutical standpoint, where there are clinical trials that start off, and we first have to have dosing paradigms to be able to establish what can be used in a safe fashion and then eventually dosed to a therapeutic fashion. And in this instance, we utilized exercise intensity for that dosage. So for the first of the 2 studies, we determined the dose response and safety of mild to moderate intensity isometric resistance exercise in children. And so with this focus on safety, we had 3 safety measures that we utilized. As a group here at the University of Florida, we have developed quite a bit of experience in use of MRI with skeletal muscle, and particularly with neuromuscular disease and Duchenne. And so our primary safety outcome measure was T2 weighted MRI. This is a construct that is utilized to represent muscle damage, inflammation, and edema in a sensitive yet non-invasive manner, and has been correlated with histological measures of muscle damage as well. And so we were able to go ahead and image the bilateral thighs of these participants so that we could have this information. And then, as you can see in the image, where we were able to then individually focus on muscles that were in the quadriceps or knee extensors, and then muscles of the knee flexors or hamstring muscles, in order to determine if there had been any signs or indications of muscle damage as we were working with these boys with this dosing. 2 other measures that we utilized were the Wong-Baker Faces Pain Rating Scale, which is a validated measure for assessing pain in children. And then, as Alan had talked about earlier with regards to creatine kinase levels, we used a blood draw to be able to assess creatine kinase levels in these participants pre and post exercise. And then for the dosing itself, we needed to find an individualized manner of doing so. And so we utilized this as a Biodex isokinetic dynamometer, where we were able to position the leg where it would be stable for isometric, or like Alan described, the no movement kinds of contractions. And we had the boys perform these isometric contractions for both the knee extensors, where they were straightening the knee or attempting to straighten the knee, as well as when they would attempt to bend the knee. So for the knee extensors and the knee flexors. And this was done at 2 different positions. We did it at approximately 30 degrees of knee flexion, or 30 degrees of the knee being bent, as well as at 60 degrees. This was important because, as we know, strengthening a muscle at one static position will not have as much functional capability or potential than if we're strengthening at different points in the range of motion. And so we had the boys perform these maximal voluntary contractions, where we provided them a lot of input and encouragement while they were kicking and pulling, as well as receiving visual input from the Biodex itself, where they had this display, which the harder they kicked or pulled, the higher the columns, to give them some other motivation and feedback as we strive to ensure a maximal effort. We then had this timeline set up with 3 different experiments. So in the first experiments, the goal was to recruit and enroll 4 boys with Duchenne muscular dystrophy that would participate in the 3 safety measures on day 1, as well as the strength assessment for MVC of the knee extensors and the knee flexors at both 30 degrees and 60 degrees knee flexion. They would then return 2 days later and do an exercise bout at an intensity level of 30% MVC. These were exercises done 4 sets of 6 repetitions for both the knee extensors and the knee flexors at both 30 degrees and 60 degrees of knee flexion. And then at day 5, they would return for a reassessment of those 3 safety measures. If at least 3 of the 4 boys did not show signs of muscle damage, the goal was to then move to experiment 2 with 4 additional boys, who would then perform the exact same paradigm, only as in a dosing type of escalation, we now increase the intensity to 50% MVC. And if at least 3 of those boys did not show any signs of muscle damage, we then would move to experiment 3 with 2 additional boys, who would perform the same paradigm as in experiment 2 for 1 week with 1 bout of exercise, but then would undergo 2 more bouts of exercise the following week at 50% MVC. So looking at repeated bouts of exercise and its impact on muscle. For our results, we did have 4 boys who enrolled in and completed experiment 1, and not only just 3, but all 4 boys were able to complete that exercise bout without any indications of muscle damage. We had an additional 4 boys who were enrolled and completed experiment 2 at 50% MVC, and again, none of the 4 boys demonstrated any indications of muscle damage. We then moved to experiment 3 with 2 additional boys who did 50% MVC for those repeated bouts. And again, neither of those 2 demonstrated any indications of muscle damage. And so here in this schematic, what I have is this is the CK levels showing pre and post exercise. So these are the results from experiments 2 and 3. On the x-axis, we have individual subjects. So these are the 4 subjects in experiment 2 who performed the isometric exercise at one bout at 50% MVC. And these 2 are the 2 in experiment 3 who performed the repeated bouts. On the y-axis, we have the creatine kinase levels. And you can see that from the black bars representing baseline and the gray bars representing post-single bout or post-repeated bouts, that there was no significant increase in creatine kinase with single bout and repeated bout exercises utilizing this isometric paradigm and parameters. So we were very pleased with these results as we went back to our original purpose of, again, exploring the safety and feasibility of this kind of exercise. And now we had some data and information that we could utilize as exercise parameters. And so from study 1, we now were able to say we have this isometric strengthening exercise program or bouts of exercise that were done at 4 sets of 6 repetitions at an intensity of 50% MVC for the knee extensors and knee flexors at 2 different positions that were safe. Now, the important element is the follow-up, right? To be able to see, well, how can this now be implemented, not just within a 2-week program, but in something that's more structured and longer. And so the second study, that was its intent, to implement a pilot home exercise intervention study consisting of isometric strengthening to examine feasibility and safety. And I will emphasize that the main focus of this portion for study 2 was not on efficacy. We'll talk a little bit about efficacy, but it really was, can this be done in a safe fashion? And is it feasible for these boys to perform this for several weeks at a time? So for the study itself, we had various measures. And first, we had, of course, safety measures, as that was our primary focus. We utilized the same 3 safety measures that were utilized in the first study, namely the T2 imaging, the pain scale, and the blood draws for the creatine kinase levels. Secondary outcomes include both strength and function. So we looked at the MVC as an assessment of strength for both the knee extensors and knee flexors pre and post training. And then we also looked at the time to ascend and descend stairs, or 4 steps, which is an outcome, as you likely are already aware, utilized it often in clinical trials as well as in clinic itself. So for this study, what we had established for a timeline is that these participants would come to the University of Florida, where they would participate in 3 safety measures, the functional measure of timed ascend and descend steps, and the strength assessment for the isometric MVC, from which we would then be able to determine what their 50% MVC level was on an individual basis. Because later in that day, they would do their first bout of exercise. And again, 4 sets of 6 repetitions for the knee extensors and knee flexors at those 2 different positions. They then returned at day 3 to repeat that exercise program. And this is now utilizing customized exercise equipment, which I will talk about soon. And they would return on day 5 for reassessment of safety measures, and then 1 final bout of exercise with us in person. We then would ship the equipment, the exercise equipment, to their homes, where they would continue for weeks 2 through 12 on a 3-time-a-week basis, performing that exercise at a 50% MVC for the first 6 weeks, and at 60% after week 6 if there were no signs or indications of any muscle damage. And so they returned to the University of Florida at week 6 and week 12 for reassessments, and were then able to proceed. And again, those reassessments focused on the safety measures, the time to ascend and descend steps for function, and the strength for the MVC isometric contractions. When we had thought about what are we going to utilize as far as a piece of equipment, we initially had the thought process of this schematic, where we would have a stable chair that would have a cuff that could be attached to the participant's lower leg so that the boy could kick and pull without movement, but it would record the force exerted because of a strain gauge that would be attached, that would then output to a laptop computer that would be able to not only collect the data that was recorded, but also provide visual feedback to the boys to motivate them for doing the exercise. And then, of course, as theory moves into practice, we looked out to see what could be done. We found a commercial device that is utilized for concentric and eccentric type of contractions for knee extension, knee flexion, and we made modifications. We took this and we made customized pads to be able to better accommodate the size of these boys' bodies. We had to cut and drill and make other changes to it to ensure that it was set up appropriately for, again, for their body proportions. And then we had a strain gauge that we were able to attach so that they would be able to perform isometric contractions as they went and they kicked or pulled against the bar, that then had an output to a computer laptop that would provide this kind of output for the boys to see. So they not only received the chair that was shipped to their homes, but this laptop went with them. And so this, what we're seeing on this laptop is real data that a participant would have seen while he was exercising. On the x-axis, this is time. On the y-axis, we have force. And these blue hills here, there's 6 of them. And that has to do with the boy who is kicking or pulling with his effort to hold that contraction for 3 seconds and rest. And then repeat that for 1 set of 6. These horizontally placed black bars, we as investigators were able to place based on the individual, which were at plus 10% and minus 10% of the expected intensity. And you can see this boy did a very good job as far as what was needed in maintaining that approximate 50% MVC. Now, not only was the boy able to see this in real time for feedback, but as you can see here, Paul, one of our research team members, and other team members, we would Skype in so that not only would the boy be able to see what was going on the screen, but we were also able to see in real time what was occurring as the boy was kicking or pulling with regards to these force time traces. And we could provide real-time motivation, instruction, and direction throughout each of these exercise sessions that were done on a 3-time-a-week basis for the 12 weeks. So for our results, we did have 8 boys who enrolled in the study. Of those 8, there was 1 boy who withdrew. His withdrawal had to do with a burden of participation and anxiety, and he withdrew in the 2nd week of the study due to those issues. We had 7 of the remaining 8, or 88% of the boys who enrolled, complete the exercise program. And of those 7, they were able to participate in 27.4 exercise sessions over approximately an 11-week duration, with a compliance rate of nearly 85% with the exercise session and a range of 75 to 96%. We were very pleased with this compliance in that in the literature, exercise programs that have a compliance rate of 75% or above are generally deemed quite successful. When we looked a little further at the results, none of the boys had indications of muscle damage throughout this 12 weeks. And so what I'm demonstrating here, this is our primary safety outcome measure, the T2 weighted imaging. And on the y-axis, we have this muscle T2 in milliseconds, or what is the brightness that can be quantified with regards to the images itself and looking at the muscles. And we have both the baseline knee extensors and the post-training knee extensors, as well as for the knee flexors. And we see that there was no significant increase, less than 3% change from baseline to 12 weeks as we looked at this T2 imaging as a safety measure. And again, reflecting that there were no signs of muscle damage. However, when we looked at strength and function, we did see changes over the 12 weeks. With regards to the peak isometric strength and looking at the peak torque, we were able to see that for the knee extensors, we had a statistically significant increase in strength, as well as for the knee flexors. For the time to ascend and descend 4 stairs for function, we had an improvement, which would be a decrease, being able to go up the stairs faster. We had an improvement of nearly a half second that did not quite meet statistical significance and had a trend of a p-value of 0.09. But we did have statistical significance with a reduction in the time to descend the stairs, which was greater than a half-second reduction. So for some conclusions from this work, first and foremost, this was a pilot study focused on safety and feasibility. It was not a randomized controlled clinical trial, and therefore the results cannot be generalized. That said, we are very pleased and excited with these enthusiastic findings that for these 7 of 8 participants who completed this full 12-week program, they tolerated the testing very well. For the protocol that was utilized, it's safe, feasible, and it had excellent compliance from the participants. And importantly, We also saw improvements for all 7 participants who completed the exercise program for both strength and function. And we are currently continuing to work and build upon what is ongoing with these exercise sessions and studies at the University of Florida. One of my colleagues, Tanya Taivasalo, has the principal investigator of 2 ongoing studies. One is an Edgewise therapeutic study that is focusing on the development of having more blood biomarkers that can be used in exercise. As both Alan and Tina mentioned, that this is an important area of consideration. We're currently looking to recruit and enroll ambulatory as well as non-ambulatory patients with DMD who are at least 6 years of age or older. And there's a second study that's a Department of Defense study that's looking at the impact and interplay of steroids on exercise, where we will be utilizing, and again, this is similar to the original device we had in the previous study, but has now been improved as we've collaborated with some engineers on campus to where we can not only do isometric strengthening exercise, but there's an attachment that can be removed that will allow for cycling or aerobic type of exercise. And so we're, again, currently recruiting boys with DMD between the ages of 5.5 and 8 years of age. And I would like to acknowledge there's a lot of great individuals who have been participating in this this work that I presented, and just really will highlight Dr. Taivasalo and Dr. Vandenborn, as well as the National Institute of Health for their funding. And just to give a summary of everything that's been discussed by Alan, Tina, and myself, skeletal muscle really is such a dynamic tissue, and there's so much untapped potential and understanding of what we have in general, but in particular when it comes to neuromuscular disease and with Duchenne muscular dystrophy. And it's important to keep in mind that this is an organ that does not act in isolation. Muscle function and metabolism impact many organ systems and disease pathways. Also, with what Tina and I have really talked about is that we have been able to find that isometric and low intensity exercise is safe. This is a shift in what has been the thought process. And so as we continue to explore what can be done and have a better understanding of these protective mechanisms in what's ongoing for the disease, as well as any modifiers that can make therapeutic benefits, not just alone with exercise, but in combination with other kinds of therapeutic, pharmaceutical, gene therapies that are ongoing and will be forthcoming in the future. However, in order to be able to best understand, we need to have better outcomes that can be sensitive in assessing the impact of exercise on pathophysiology and DMD. And we're fortunate to have further work that's ongoing both at Stanford University and at the University of Florida to continue to investigate this area. And I'll just put up here, this is some contact information for Dr. Taivasalo at the University of Florida, as well as Dr. Duong at Stanford University. And we would be very excited and enthused to hear more from you with regards to questions, with regards to inquiries, as clinicians, scientists, and of course, most importantly, as families. As together, we not only join in this fight, but continue in the fight to end Duchenne with the Parent Project Muscular Dystrophy. Thank you very much.